Good morning. This is a podcast on the chapter 9, Cellular Respiration, and it is going to be a lengthy podcast, lengthy lecture, so brace yourselves for that. And we move to the next slide. Okay, normally by the second or the third slide, I sort of sum up what are all the different topics that you should know about this chapter. It is a good idea. It sort of lets you know. You can go back and see, oh, okay, oxidation. Do I know about this, or what is, and how is the stepwise manner process, and what is glycolysis, and what is Krebs cycle, and so on. Okay. Now, this is a question I normally pose my class, and it's a simple one, albeit. I would say, what do you do when you breathe? And very easily, people end up saying you breathe in oxygen and you breathe out carbon dioxide. So then my next question would be, you have breathed in oxygen, but you're breathing out carbon dioxide. Where did this carbon come from? So eventually, when they think back, it boils down to the fact that it is coming from the food that you eat. Okay, so okay, so we move to the part where, okay, so now let's sort of sum up this process and see how it goes. So you are hungry. You are hungry, okay? Bear with me, I'm trying to understand this particular system of recording. I apologize. So, when you're hungry, what you do is you eat. So, what you eat, let's assume it is a burger for now, okay? So, burger, what are all the things that it might have? It will have some bread, it might have some meat, it might have some other sauce, lettuce, and so on and so forth. Okay, sauce, lettuce, and so on and so forth. Okay, so now this bread, it is composed of starch, which is your source of carbohydrates. Okay. Okay, so this carbohydrate that is present gets digested into smaller molecules. Okay, now the smaller molecules, a unit of that, the best unit that is taken in easily by the cell is glucose. So you might ask, how is meat going to get into it? How is any fat or butter going to get into it? So to understand that, let's look at the next page for a minute. Okay, so here is the thing. I would like for you to imagine that there is a race that's starting kind of, once again. There is a race that starts at this particular point, okay, this point, and from there you have a certain spot, and then a final destination. So assuming that you are starting from place X, you go via a certain road, like a 5K run. Imagine there is a 5K run, and you go from this place, you pass through a certain important point, and then another point, and another point, and then you reach the final destination. That makes your 5K. Now, an old person like me is not likely to start from here, but I would like to participate, so probably I would join the race somewhere here, or somewhere here, or some people might join it here and then still feel the satisfaction of having completed the race. In other words, the cell has a certain pathway through which it goes through and a certain mechanism by which it is making energy. This particular mechanism is the only mechanism that is the most efficient system the cell has, and this is what it uses. Most of the cell, let's put it this way, 90% of all cells probably use this method to gain energy. Now, with that in as our background, if your starting point for getting energy is glucose, so you sort of start from here and it goes through a series of steps wherein it forms a lot of intermediates, and these finally I end up giving out carbon dioxide, which is my end point. So I have like completely broken down my glucose and I am deriving energy out of this. This is the pathway that a cell uses. Now, supposing the name of an intermediate, for example, I would like to throw in a name. Let's call it acetyl CoA. Now, this is an intermediate which sort of is formed at this step and then enters the cycle or the process of breakdown at this particular point. Now, if I have like a fat or a fatty acid can be broken down and I can get a smaller intermediate from that, and that is acetyl CoA, which means a fat can be broken down to give me acetyl CoA, and acetyl CoA can join this energy deriving pathway at this particular point. And from here, I can break it down further and derive whatever energy that I get from it. So, it is just possible it avoids all these steps, but it does join the main pipeline or the main course at a certain point. Similarly, other products or other materials that are present, like your protein or any other sugars or any other large form of fat, would probably join this particular pipeline or pathway of deriving energy at various points, and the cell can still manage to extract energy from them. That is the point I was trying to get. Okay, so let's move to the next. Now, what are the different kinds of activities that would need energy? Running, dancing, cycling, swimming, walking, dancing slowly, driving a car, sitting. All of these would need energy. And this is specified in the terms of kilocalories. One second. In terms of kilocalories, and that is what is specified with these values. So these values are kilocalories of energy required to do a certain thing in a certain speed. So that is what is the body's requirements. Now, where am I getting this energy? How is whatever I need moved on from the food that I eat to finally the ability to perform a certain action? How does that translate to that kind of system? Okay, so how we go about explaining this whole process is you have let's just divide this whole process. Now, you and me. Okay, so now what we do is we consume food. Okay, we respire and breathe in oxygen. We have this huge big powerhouse with us which is present in all of the eukaryotic cells. So we are all eukaryotes, by the way. So we definitely have plenty of mitochondria in our system. And it has the setup, the mechanism to derive energy. And all of that energy is derived from the food and turned into ATP. So this chapter in a nutshell is basically how glucose combines with oxygen and how this mitochondria goes through a sequence of steps to derive ATP from this, and how we let or completely break down this glucose into carbon dioxide and water molecule, right? That is what is this chapter all about. Okay, now I would like to talk about the process of the hill. Okay, so imagine, let's go back. Imagine that there is a hill, and this particular hill has water at the top. Now, if there is water at the top, what is the normal expected course of action with the water? The water will flow down. Now, did you put energy into it? No. You didn't put any new energy input. Okay, so you didn't put in any energy for this process, but it came down. Now, then the water collects itself at the bottom. Now, can this water go back up on its own? No. But you can pump it up. Now, when you pump it up means you put in energy and then bring it back up to the top. Now, when this flows down with great speed, great force, what can happen? Imagine this. You have a turbine with blades, okay, and you make a mechanism, kind of a setup, wherein the water flows on, and the movement or the force with which the water is flowing is capable of turning this turbine, turning the fans, and that generates mechanical energy. And I can convert this mechanical energy into electrical energy. And this is the basis on which I get my light in my house. I can switch on light because I have electricity. So this is the way I can make use of the presence of water merely because of its presence on top of a hill, wherein its ability to flow down releases energy. And this release of energy can be used by us smart, if we are smart enough, if we set up a mechanism to tap this resource and then convert it into a usable form of energy. Our body makes use of this very same principle in deriving energy from glucose. Going back, let's see. Okay, going back. Now, think about it like this. Glucose that is present is a high energy molecule. It's a high energy molecule. Why? Why does it have a lot of energy? Remember, the structure or the molecular formula of glucose is C6H12O6. Now this has got a lot of hydrogens, or in turn it can be explained as a lot of reducing power, or a lot of electrons or protons, as you can explain. Okay, so because of this particular molecular formula that it has a lot of potential for releasing that energy, or rather, put it in a different way, the cell has the ability to extract all of this and make use of that in another pattern, which you will see in a while from now, where or what is the fate of all of this H. But in the end, what do you get? You get carbon dioxide and you get water. Okay, and here you have glucose plus oxygen. If you notice, this particular glucose had a lot of protons in it. It had a lot of hydrogen in it. And if you notice, the carbon dioxide, which is what it turns or becomes into, that has practically no hydrogen at all. And it is this oxygen that gets completely reduced to water. So glucose gets oxidized to carbon dioxide and oxygen gets reduced to water. Okay, this whole process results in release of energy, and this whole process is also exothermic. Now if it is so, I can tap that and get my ATP. And how, we will see in a few series of steps. So this is in a nutshell what happens inside the process of respiration. Okay, so when you go back to read the whole process, in fact here, one second, bear with me. When you go back to look at this process, this is your respiration. And this carbon dioxide and water, or H2O, is then used by plants in presence of sunlight and turned into glucose. And that is your process of photosynthesis. So if you look at it very, very carefully, the whole reaction, the process of respiration involves the breakdown of glucose with the help of oxygen to release carbon dioxide and water, and this is a energy released out of this. Okay. On the other hand, photosynthesis is the putting together of smaller molecules of carbon dioxide and water together in a form wherein it would become a high energy molecule, but you need energy. That energy is derived from the sun's light energy. This process requires energy. Okay. Okay. So that is what this particular page says. Okay. Now, just because I have heat, I cannot. Let me go back. Just because I have heat, I cannot move the flagella. I cannot pump solutes across the membrane. Remember, in active transport, I needed energy to move certain solutes from one side of the membrane to the other side. And then I also needed to polymerize monomers, meaning I had one monomer and another monomer. These two guys are joined together, but I need ATP for this whole process. Now I'm using ATP to join them together, and if I want to add one more, I sort of need ATP for this whole process. Okay. Now perform other cellular work. What kind of cellular work? Like cell division, mitosis, that is, okay, or movement. How do I get these things done? Okay. So all of the breakdown of high energy molecules, which is part of what respiration does, meaning glucose being a high energy molecule is broken down. Okay, that is called catabolism. Okay, so catabolism, I would, for this whole process, if I'm breaking down a high energy molecule, I get ATP. Okay, now I cannot keep on getting ATP just out of thin air. How do I get my ATP? I need to have ADP and PI. Let's just look at what is this ATP. Okay, this is adenosine, okay, which means it is adenine, plus ribose sugar plus 3 phosphate groups. That means adenosine triphosphate, 3 phosphate groups. Okay, so this is not made out of thin air. This can be made only if I have ADP and an inorganic phosphate readily available, and I can put it together and make it into ATP. Okay, so let's revisit this whole thing. Now, catabolism. Catabolic activity. Okay, that means breakdown of glucose. Okay, what will happen to it? I will release energy. So, what will I get? If I am releasing energy, I will get ATP. How did I get this ATP? I had ADP plus inorganic phosphate put together by this catabolic activity, and I make ATP. Now, I have anabolic processes. This means I am building stuff, or biosynthetic synthesis, or building stuff. For that, I use ATP. When I am using ATP, I convert it back to ADP and inorganic phosphate. So, it has to be remembered, ATP is not made out of thin air. It is made only if ADP is present with inorganic phosphate. Then these two things are put together and I get ATP. Now, in an anabolic processes which requires energy, I need the ATP. So if I have taken the ATP, meaning what I basically do is I have the adenine, ribose sugar plus the 3 phosphates. It is the last phosphate, if it is broken down or removed, that particular bond releases a lot of energy. And so I use that by breaking that particular bond, breaking the ATP, I use that energy to drive all my anabolic processes, which means I make it or reconvert it back into ADP and inorganic phosphate. So the whole process involves a kind of a coupling mechanism wherein anabolic processes are in synchrony with catabolic processes, and ATP is always regenerated back by the smaller products, the ADP and inorganic phosphate, which again is broken down to give me ADP and inorganic phosphate, which is put together to give me ATP, and so on. So this cycle will keep continuing, wherein I will have a certain fixed amount of ADP and inorganic phosphate in the cell, which the cell can put together to give me ATP. I use that and then I make the smaller pieces available, and these pieces are put together again to give me my energy, and so on. That's the way it works. Okay, so breakdown of glucose essentially leads to a forward, like a reaction that goes forward on its own, and it is exergonic, it is spontaneous, it has a negative delta G, and it can take place without input of energy. Okay. Now, this particular picture, what it basically tries to tell you, I would like to give an example of how you would do a certain test, or a certain, like an analogy. The analogy is like this. If you have a potted plant, so you have a potted plant, okay, at the bottom of your house and you are about 3 stories above, living above. Now you want to water this plant. You cannot possibly take one huge big tanker and throw all that water onto it in order to water it. What would happen if this were to be done? It would get watered, but the impact would literally make the plant die, which means this is like I am watering it, but this is not the amount. Instead, if I would go through a process where I pass it on to someone, then that person passes it on to someone else, then that person passes it on to someone else, and then finally I'm able to add a small 2 mug full of water into it. That way I am keeping the plant alive, plus my plant is getting the water. So if you go back to think about this particular picture, what I'm trying to say in this is hydrogen and oxygen, when they combine, it is an extremely explosive reaction. Now what do I mean by this? Remember, it was C6H12O6 plus I have oxygen and I get carbon dioxide and water. Now how does this water happen? This oxygen combines with some amount of hydrogen to give you the water, right? In the end, that is what this translates to. Now if I let just oxygen and hydrogen combine together, it is an extremely explosive reaction. It will give me water, but that kind of release of heat and light energy is way too much. That does not help me. That is not possible for me to tap. It is just lost into the atmosphere. Now, I cannot burn up my cell every time by making them mix in this kind of uncontrolled fashion. I will not live. I'm not a living creature anymore. Okay? I'm just a fried up piece of whatever, right? So instead, the cell does the same process of combining this oxygen with hydrogen to give you water, which is what it gives, but in a very smart way, in a series of small steps. Okay? The small steps involve a series of steps that would be listed out in the next page, please. Okay, so here is what you have. Now, glucose, okay, when it starts, it goes through a series of steps, 10 steps. It is called glycolysis. And then it goes through another set of steps called Krebs cycle. And then it goes through another set of steps called electron transport chain plus chemiosmosis. Okay, now what it basically does is this glycolysis is 10 steps. This is another set of steps. So a small set of steps. Okay, this means at every step it is able to, every other step maybe, it is able to extract some energy. It sort of collects them all as energy packets. Okay, kind of you ask for donations, small bits and pieces of donations, and you put it all together, and then finally you get one huge big lump sum. And then you say, oh, I have the money to build this swimming pool or this recreation center or so on and so forth. Okay, so that is how the cell behaves. It goes through a set of series of small steps, and these small steps help the cell extract energy at different points and then collect all of that energy and go to a certain part and bank it and encash it. Okay, so then you would see that you have to you will observe rather that the cell has these components undergo a series of reactions, and they could be oxidation reactions or reduction reactions. Okay, sometimes it is in such a way that oxidation and reduction reactions are sort of combined. They are sort of in a same place or happening to the same molecule sometimes, and it's sort of at a certain point it is oxidized, at a certain point it is reduced. Now, how do you understand if a certain compound is oxidized or a certain compound is reduced? A nice way to remember is oil. Oil means it loses electrons, loss of electrons. Rig means it gains electrons. Okay, and that would be reduction. Okay, so in this particular case, if you see, glucose is already reduced. Okay, it loses all the hydrogen atoms. Now, a lot of you might question this. Sometimes I'm saying loses electrons, sometimes I'm saying loses hydrogen. Now you see, the important thing is electrons cannot travel on their own. So if I'm saying losing electron means it is losing a proton. That's what it means. Okay, so it loses all of these hydrogen atoms. Okay, and that makes it get converted into carbon dioxide, which is completely oxidized. Okay, so from being reduced it has become completely oxidized. On the other hand, oxygen is very oxidized and it gains the hydrogen atoms and becomes reduced. So you see, in a nutshell describes both a reduction reaction and an oxidation reaction. Okay. Now, organic molecules, most organic molecules have a lot of hydrogens in there. One second. A lot of hydrogens present in them, and so they are called hilltop electrons, or like water on top of the hill. Okay, and they are in a high energy state, meaning they are very, very unstable and ready to lose it. Okay, and so they transfer the electrons and then they get converted into a low energy state. But now put it in perspective. When you just have glucose in a spoon full of glucose, like a gram of glucose, and you burn it, okay, when you burn it, it will release 686 kilocalories of heat per molecule of glucose. Now that is way too much heat. Your body temperature is only 37 degrees centigrade. Okay, there is no way your body temperature increases to accommodate this kind of heat. You won't be able to keep all your enzymes intact if it burns and heats up in this fashion. So obviously your cell has a certain system in which it is able to adjust or manage this complete breakdown of glucose into carbon dioxide and yet trap the heat into usable form and stay alive. Okay? So, once again. Okay. Now, before I move to this chapter, this part of the chapter, I would like to go back to this particular page. Okay, so here is this. Let's go with an analogy. First and foremost, before the analogy starts, I would like for you to remember two things. The first law of thermodynamics. Law of thermodynamics. Meaning, energy can neither be created nor destroyed. It can be only converted or transferred into one form or the other. Energy neither created nor destroyed. only transformed, right? Okay. On the other hand, the second law of thermodynamics says energy, whenever there is a transfer, okay, from one form to the other, there is loss of energy as heat. This is something which is unavoidable. You cannot avoid this particular situation. Okay, which means what you have to look at this particular point is, how is it even possible that the cell, from the step of glucose, going through so many series of small steps, it is able to keep the energy intact? How? Okay. In order to understand that, I would like for you to think about a situation. Imagine a particular situation wherein you are in a football game. Okay, in a football game, wherein in a football game that you are in, you have a player who has fallen and injured himself. He has injured his leg. So I have this person running. Because there is no first aid kit, she runs to a restaurant nearby to bring ice in her hands. This another person runs to the household or something nearby and tries to bring some ice. And this third person runs to the vending machine or some place with an ice box. Okay, so now this person brings it in his hands. By the time he comes here to give the ice, it has melted. So he has lost it. He brings it in a vessel. By the time he comes back, it becomes water. It is not very usable. This person brings it in a box, an ice box. That is kind of like a cooler. It is this particular person who is able to bring the ice intact. Now, what is the analogy between this and what I spoke to you some time back? In your body, you have a particular chemical that goes to almost every step of these reactions, and extracts the energy and kinds of puts it in an ice box. And when the compounds transfer their energy to this particular compound called the NAD plus, there is minimum loss of energy. So it is a smart thing to transfer all the energy that it gets to NAD and turn it into NADH plus H plus. Okay. So basically what he's trying to say is, it is transferred not directly but in a redox reaction, wherein if it is able to collect the electrons / the protons, it becomes NADH and H plus, and this is like a energy rich molecule. Okay. Electrons lose very little of their energy when being transformed to NAD+. Okay. Now, each of this represents a stored form of energy. So, what the cell basically does is, during the series of reactions, it transfers it to NAD, transfers it to NAD, transfers it to NAD. And then I have a lot of this NADH, and it is like, I am promised, yes, in the end, I will get energy from it. It's like a check. It's very valuable, right? And then what happens is, at a certain point, they would reach the electron transport chain. And this particular electron transport chain involves a process wherein the electrons are transferred to the carrier. One second, let's go back 1 minute. The electrons are transferred to these electron carriers. So this NADH finally comes, a lot of them finally come, a lot of NADH is present. They come and transfer it to this guy. He transfers it to this guy. He transfers it to this guy. He transfers it to the next, kind of like that. So whenever this transfer happens, there is also another thing that happens, okay, which we will go into in a while. But this process is the one which leads to the formation of water and leads to the formation of ATP. Okay? So the stages of cellular respiration are divided into three main parts. Okay. So the first step is glycolysis. Glycolysis means breakdown of glucose. Okay. Now in this step, please mind it. It is only breaking down glucose. Kind of, if you have like a huge piece of bar of chocolate, think about it like that. You've just broken it down into a smaller, two smaller bars of chocolate. That's what has happened at present. Okay, let's see. So, think about it like this. This is your entire cell. Okay. Now, in this cell, it has a mitochondria. A brief story about the mitochondria. One, remember, all cells which are eukaryotic will have mitochondria. And there are many mitochondria present in every cell, all right? It's not just one of them, and it's not. And the way it is structured is, this is the outer membrane, this is the inner membrane, and this is the intermembrane space, and this is the matrix, mitochondrial matrix. Okay. Now, glycolysis takes place in the cytoplasm. So the glucose has entered the cell and glycolysis is taking place here. It goes through a process of 10 steps. Okay, at the end of it, I get 2 pyruvate molecules. Okay, and so glycolysis means what? Glucose, right? And glucose is a 6 carbon. Okay, so 1 6 carbon molecule, 1 glucose molecule, okay, gives me 2 pyruvate molecules, and each of them is a 3 carbon. Okay, so this is what I get at the end of glycolysis. I also get a few other things. 1, I get 2 NADH plus H plus, and I get 4 ATP. But, very important, but for kick-starting the process of glycolysis, I needed 2 ATP to kickstart. Like an investment. Because I used up 2 ATP, my net ATP is only 2 ATP. Okay? So, one more time. Glycolysis occurs in the cytoplasm. It does not need oxygen. It is marks the beginning of cellular respiration. Okay? It breaks down glucose, which is a 6 carbon, into 2 3 carbon molecules called pyruvate or pyruvic acid. Both are same names. Okay, now that I have that, so I have, let's draw this. One 1 2 3 4 5 6, and then let me call this, okay, one kind. Excuse me. So now I have them as, sorry, excuse me, 2 3 carbons, right? So then this one pyruvic acid, okay, is a 3 carbon, mind you, it has to get into the mitochondria. Okay, this is the mitochondria. So it gets into the mitochondria. Once it gets into the mitochondria, here it cannot enter the cycle. Okay, it has to get converted. What does it get converted into? It gets converted into acetyl CoA. What is acetyl CoA? It is a 2 carbon molecule. Now this 2 carbon molecule means from 3 carbon it has become 2 carbon. Where is the 1 carbon? It is lost as carbon dioxide. So there is 1 lost. Alright. So this is a 2 carbon molecule. So this enters the cycle. Now I want you to go back and think about 2 different things in this. Okay. One is, have you ever seen people in a carousel or a merry-go-round or carousel, right? So you have this carousel which keeps turning. So there is some people who can get in, there is some people who can get out. Do you agree? But it keeps turning. That is always there. Okay. So the cycle is constant. There are certain components in the cycle that are a constant. You cannot change it. It has to be present. Okay. So there is a 4 carbon compound that is constantly present in that. So a 2 carbon compound, that is the acetyl coenzyme A, enters this cycle. So it becomes a 6 carbon compound. It loses 1 carbon dioxide. It becomes a 5 carbon compound. It loses another carbon dioxide. It becomes a 4 carbon compound. And that gets restructured back, ready to accept another one. Now, tell me, have I lost all the carbons? So first I had, I had 1 carbon dioxide lost, 2nd carbon dioxide lost, 3rd carbon dioxide lost, right? Now, have I completely lost all my glucose? Have I? The answer would be no. Why? Because my glucose is made up of 6 carbons. Okay. Now these 6 carbons have to be accommodated for. At present, with 1 turn of the Krebs cycle, I have only lost 3 carbons. I still have to account for the other 3. Remember, I have 2 3 carbon molecules, 2 pyruvic acids. So 1 pyruvic acid gets converted to acetyl coenzyme A and loses 1 carbon dioxide, and then enters this, loses the other and loses the third. So the 2 carbons that entered got lost through the end of one turn of the cycle. The other pyruvic acid also has to do the same thing. Okay. Now, this citric acid cycle that I'm talking to you is a very significant cycle, and it is got other names also. One, it is called citric acid cycle. It is called Krebs cycle. It is also called TCA cycle. Okay, so it takes place in the mitochondrial matrix. Okay, it oxidizes the pyruvate into acetyl coenzyme A, which is a 2 carbon. This enters the TCA cycle. It applies the 3rd stage with electrons. Now, at the end of citric acid cycle, citric acid cycle, what do I get? Okay, so my glycolysis, I had so this is my glucose to 2 pyruvic acid, 2 NADH plus H plus 2 ATP. Okay. Now this pyruvic acid gets converted in an intermediate cycle. Pyruvic acid gets converted to acetyl CoA and I get an NADH. If it's 2 of them, I'll get 2 of them, and I also lose. Clear? Okay. Now it enters the Krebs. When it enters the Krebs, what do I get with 1 turn of Krebs cycle? 1 turn. I get 3 NADH plus H plus 2 CO2, 1 ATP and 1 FADH2. This is what I get with 1 turn. But my entire glucose is not completely finished, burnt up, lost with 1 turn. I need to do 1 more turn. So 2 turns of Krebs cycle would give me 6 NADH, 4 CO2, 2 ATP and 2 FADH2. So at the end of Krebs cycle, what do I have? Let us just look at it. I have 6 CO2. Okay. I have 2 NADH here, 2 NADH here, that is 4 and 6. So 10 NADH plus H plus and 2 FADH2 and 2 ATP here and 2 ATP here. 4 ATP. Okay, so this is what you get out of the whole process of complete breakdown of glucose, and this is the number of NADH and H plus and 2 FADH2 that you get. What is this NADH and H plus? Okay, it is called nicotinamide adenine dinucleotide. Okay. Okay, so the last stage is when all of the when all of the NADH and the FADH2 now walk towards a certain place. Okay. So I would like for you to think of that as, if you watch Pirates of the Caribbean, you have Bill Turner, who's dead, and then all of the fish people, zombie-faced people in Davy Jones' ship, they all start walking towards Bill Turner to carve his heart out. They start saying, part of the ship, part of the crew, part of the ship, part of the crew. So all of them are walking to a certain stop. So think about all the different NADH that is formed in different steps of the different reactions. Okay, they are all walking towards a certain spot. Okay, and that is the main part of the third step. Okay, so let's see what happens. Okay, so I want you to be able to imagine the mitochondria in such a way that you are able to see that the mitochondria has an outer membrane and an inner membrane, right? This inner membrane has a lot of electron carriers. Now these electron carriers, when they have an electron, they are reduced. When they lose an electron, they are oxidized. So they sort of switch or toggle between the two situations, being reduced or oxidized, reduced or oxidized. So every time an NADH would come and give the electron to the first carrier, he would become reduced. When he gives the electron to the next fellow, he himself gets oxidized and the next fellow gets reduced. Okay, and it will keep happening throughout the electron transport chain. Okay, let me just go back to a certain blank page if I have one. So it's kind of like this. If I have them all arranged in this fashion, okay, normally if I were in class, I normally ask my students to simulate this. So I have the electron carriers in this top. In the end, I have oxygen waiting. Okay, so what happens is the NADH transfers the electrons to the first guy. Okay, so now this first guy gets to be, because he is gotten the electrons, he is reduced. Then what he does is he transfers the electrons to this guy. So this guy gets reduced and this guy gets, once again, oxidized because he has lost it, and it sort of toggles between being oxidized, being reduced, being oxidized, being reduced, and so on, back and forth, back and forth. They sort of just toggle between these two states. Okay, every time one electron carrier passes the electrons from itself to the next, there is a small amount of energy. Okay, and with that energy, what they do is they are able to throw out a proton. Okay, so this transfer, throw out a proton. This transfer, throw out a proton. This transfer, throw out a proton. So suddenly, with this NADH in picture, there is a lot of protons in the intermembrane space. Okay, finally it reaches the end of the electron transport chain where oxygen is situated very conveniently, and this oxygen, when it collects the electrons, it takes the protons from the matrix, okay, and becomes water. So the whole story is over. What did I get out of this process? I got my carbon dioxide completely gone, all my NADH and FADH2 came and donated their whatever electrons they had to the electron transport chain, and at the end of it I had water being formed, oxygen reduced water being formed. But I also have something. There is a gradient. I have a lot of protons in the intermembrane space in comparison to the inside. So there is a high concentration of protons in the intermembrane space and a low concentration inside. What is going to happen? It's wanting to come back in. Remember, high to low, like a passive diffusion, right? Okay, like passive transport or a diffusion. Okay, now immediately adjacent to this electron transport chain, there lies a certain enzyme called ATP synthase. What is its claim to fame? It helps in synthesis of ATP. It cannot make it out of thin air. ADP and inorganic phosphate is just present close by. So what now? All of these protons try to find their way back inside. They flow down, they come gushing down inside, and this creates energy. And with that energy, they are able to convert ADP and inorganic phosphate to ATP, and I get ATP. This process is called chemiosmosis. Okay, so it shuttles the electrons. The third step is the oxidative phosphorylation. It shuttles the electrons to the electron transport chain, which is embedded in the internal mitochondrial membrane. And this involves chemiosmosis, wherein it is movement of protons from high to low. Okay, it is a special process discovered by a scientist named Peter Mitchell. Okay, generates ATP through the process called oxidative phosphorylation, and it is associated with the process of chemiosmosis. Okay, so in a nutshell, let's just sum it up. I need to also talk to you about what this particular picture means. Let me just go back to this. Okay, so you have your glucose being converted to pyruvate, and I made ATP. Now this ATP was made how? Just because there was an ADP and an inorganic phosphate. They were there was an ADP and there was a substance which had a phosphate group, and it transferred the phosphate group to this guy and made it into ATP. This substance, example, glucose 6-phosphate. Now this is a compound which contains a phosphate group. So if I have ADP, it can transfer this phosphate group to this guy and make it into ATP. This form or method of making ATP is basically called phosphorylating. Okay, or addition of a phosphate group. Okay, phosphorylation. Now this process wherein a substrate with a phosphate group is able to donate its phosphate group to an ADP and make ATP is called substrate level phosphorylation. You would see this in glycolysis and in citric acid cycle. On the other hand, you have another mode of forming ATP called the oxidative phosphorylation, which I explained in the slide before. Okay, so let's just go over the whole set of slides quickly. In glycolysis, a single molecule of glucose is cut through in a series of steps, okay, and 2 molecules of pyruvate are produced. 2 molecules of NAD is made, reduced to 2 molecules of NADH, and a net of 2 molecules of ATP is produced. So this is what, if you have a compound that is glucose, it has got 6 carbons, it is going through a series of steps. This is what I get out of the whole process. I get 2 pyruvate molecules, which is 3 carbons each, 2 NADH and 2 H plus, and 2 ATP. Okay. Now, for this whole process to start, it is divided into 2 parts. It's called the energy investment phase and the energy payoff phase. In the investment, it consumes 2 ATP molecules, which is used to kickstart the whole process or energize the glucose molecule to start the whole process. In the payoff, what happens is I get 2 NADH molecules and ATP molecules are also generated in the process. Okay. Now, so far I explained what is substrate level phosphorylation. This is again pictorially explaining to you what the whole process is. So I have a substrate which has a phosphate molecule at the end. It transfers it to ADP in presence of an enzyme and makes ATP. This is very limited. It is limited the amount of this substrate available. If I have a lot of this available and a lot of this available, only then I can make this. I cannot make this out of thin air. I cannot make it if I have less of this. I cannot make a lot of this if I have a less of this. That is not possible. So this is very, very limited. I cannot make a lot. Okay. So in the investment phase, you have this happening and you have a series of 10 reactions taking place, and these are the intermediates that are formed. For my class, I am concerned with the end product that you're getting. That is what you're supposed to remember in my class. Now, this is the Krebs cycle in a nutshell. Now, acetyl coenzyme A enters the Krebs cycle. Now, remember, acetyl coenzyme A is a 2-carbon molecule. Okay, it has a 4-carbon molecule already. The citric acid cycle. It's called oxaloacetic acid. Okay, and that becomes a 6-carbon molecule. It releases 1 carbon dioxide and becomes a 5-carbon molecule. It releases another carbon dioxide and becomes a 4-carbon molecule. And that is something called malic acid. Okay. Now again, this can restructure itself back to give me oxaloacetic acid, which is ready to take the next acetyl coenzyme A and start the whole cycle one more time. Okay, so per turn of the Krebs cycle, I get 2 carbon dioxides out, 3 NADH and H plus, 1 ATP and 1 FADH2. Okay. And if there are 2 turns of citric acid cycle, to explain for the complete breakdown of 2 molecules of acetyl coenzyme A, I would get in the end 2 ATPs, 6 NADH and 2 FADH2. Okay, so that's what he's trying to say in this. So oxidative phosphorylation involves the electron transport and chemiosmosis. It requires an adequate supply of oxygen. So if you actually notice, in the entire process of glycolysis, and in the entire process of Krebs cycle, we never came across oxygen. Oxygen plays its role in the last part of the breakdown of glucose in cellular respiration, in the oxidative phosphorylation, especially at the end of electron transport chain. So you have electrons from the NADH and FADH2 travel down the electron transport chain to oxygen. Then the oxygen picks up the protons to form water. The energy released is used to pump the protons from the mitochondrial matrix into the intermembrane space. In chemiosmosis step, the protons diffuse back and through the ATP synthase, which is called the world's smallest rotor, and it drives the synthesis of ATP. Okay, kind of like this. Now, there is something to be noted. NADH starts to donate its electrons, whatever it had, to the first electron carrier. So, because of that, it is responsible for a certain number of protons going out. X number of protons going out or thrown out. Okay. On the other hand, FADH2 does not start at the first one, it starts somewhere in the middle, which means it is responsible for a little less protons being thrown out. How does that translate? What difference does that make? So, the number of protons thrown out on account of transfer of electrons by NADH is slightly higher. So when they all come back inside, gushing in, the amount of ATP that can be made because of that is roughly calculated as 3 ATP. Okay, and that means 1 NADH is responsible for making of 3 ATP, whereas 1 FADH2 is responsible for making 2 ATP. Okay. Now, there are certain cellular poisons that can obstruct the process of oxidative phosphorylation. These poisons can block the electron transport chain at certain point, which means what happens is there is no transfer of electrons. So that means there is no throwing out of protons. So that means your oxygen is not being used. Okay. Or you can have the other part of the oxidative phosphorylation. For example, the ATP synthase is inhibited. So I am not able to make ATP. Okay. Or there is the third kind wherein the membrane becomes leaky. See now, what was the main way in which at the end of all of this chemiosmosis and oxidative phosphorylation, we had a difference of number of protons on the outside versus on the inside. And this gradient that we formed is responsible for, or is the driving force that helps us derive energy. Now if I make the membrane leaky, there is no gradient because they are all flowing back and forth. There is no control, there is no high, there is no low, which means it is uncoupling the whole process, meaning all the process of transferring the electrons becomes useless. Okay. So you have a poison called rotenone, okay, which stops the electron flow or transfer at this particular point, or cyanide and carbon monoxide block it at this particular point, whereas DNP is an uncoupler which sort of breaks down the membrane and makes it leaky. On the other hand, oligomycin, which is a antibiotic, it sort of blocks the ATP synthase, so then there is no ATP being formed in the whole process. Okay. So, what is the total amount of energy that you end up getting in the whole? If we actually put together everything that we have in glycolysis, I had 2 ATP, 2 NADH, intermediate stage, I had 2 NADH plus H plus. In the Krebs cycle, I had 2 ATP, 2 FADH2 and 6 NADH plus H plus. What does this translate to? 4 ATP, and these ATPs that are being formed are due to substrate level phosphorylation. Please be mindful of that. Now, how many NADH do I have? 10. That is the 2 here, and 6 here. 10 NADH. And how many FADH do I have? 2 FADH2. In 2 slides back, I explained 1 NADH is responsible for formation of 3 ATP. So 10 of them would be 30 ATP. Okay, 1 FADH2 is responsible for making 2 ATP, and 2 of them would be 4 ATP, which means 4 plus 30 plus 4, which is 38. Under extremely ideal conditions, with everything going the right way, 1 molecule of glucose can yield a total of up to 38 molecules of ATP. And this is the best the cell can do. This is harnessing only 33% of all of the energy that is possible, but this is the best that the cell can do. Okay, 34, I'm sorry. Okay, now this is in a nutshell what happened. Okay, now we move on to another part of this chapter called fermentation. Now, all of the story that we had some time back, all of that depended on when or if oxygen is present, if there is mitochondria present. Now, there are some organisms. One, they are capable of life without oxygen. How do they make their energy? How do they manage? Okay, there are some guys who, if there is oxygen, they will form aerobic pathway. If there is no oxygen, they will form the anaerobic pathway. And these are called facultative. Okay, anaerobes. There are some who they don't care if there is oxygen. They don't use oxygen. They are anaerobic. Okay. Now, there are others who die in presence of oxygen. These are obligate anaerobes. How do each of these guys obtain energy? Okay. So, fermentation. Now, in this process, what happens is there is only glycolysis happening. So then I have 2 ATP and I have 2 NADH. Now there is no oxygen. If there is no oxygen, there is no Krebs and no oxidative phosphorylation, no electron transport chain. So now I have a lot of this. What will I do? I have to recycle and get it back. Otherwise, how am I going to do the next one? So the next path involves kind of next page will tell you. 1 second. So in this particular situation, what is happening is glucose. This is the 6 carbon molecule. Okay, and I am going through, or the cell is going through glycolysis. This happens in lactic acid bacteria. Okay, and when they, at the end of glycolysis, obviously I have 2 pyruvate and I have 2 ATP and I have 2 NADH. So then these pyruvic acid molecules again take back these guys, the 2 NADH, and restructure themselves to form lactic acid. This is essentially the mode of life for lactic acid bacteria. They really don't care for the presence of oxygen. They would follow this method because they have an enzyme which does this. Lactate dehydrogenase. Okay. On the other hand, you have yeasts. Okay. What do yeasts do? Yeasts are single-celled fungi. They go through they are technically eukaryotes, right? So they have mitochondria. So how do you trick these guys into making alcohol for you or making wine for you? How do you trick them? Basically, you would not have any oxygen available. Why? If there is oxygen available, it's very easy. 1 glucose and I will easily get 38 ATP, right? But if I have no oxygen, 1 glucose will only give me 2 ATP. So if I want 38 ATP, what will I do? I will take up a lot more glucose and I'll keep converting. That means the cell has to do extra work. But here it doesn't have a choice. It will keep doing that extra work. So for that amount of energy that it wants, it will consume more glucose. It consumes more glucose and I get more alcohol. Okay. How does that happen? So here I have glucose which is going through the process of glycolysis. I get 2 ATP and I get 2 NADH and I get 2 pyruvate. Now I lose 2 carbon dioxide molecules and I get 2 ethanol molecules, meaning 1 pyruvate will give me 1 ethanol, and this is a 2 carbon molecule. This was a 3 carbon molecule. One of them is lost. So if it is 2 pyruvates, I'll get 2 ethanols, and I use this NADH to restructure my ethanol molecule. Okay, and this carbon dioxide that is released is responsible for the bubbles in champagne, or the softness that you see in bread. It's like airy bread. It's got fermented well, and you can see that. Okay. Obligate anaerobes are poisoned by oxygen and they require anaerobic conditions. They live in stagnant ponds or deep soils. They use other forms of obtaining energy. So they would have their glycolysis, their Krebs in a different way because they don't have mitochondria. These are bacteria. Okay, so no mitochondria. Okay, so their membrane itself serves as the site where all the electron transport chain takes place. So at the end of the electron transport chain, they have, say for example, a sulfate molecule, and that gets converted to hydrogen sulfide, and that is the one that accepts the electrons. So that's one way. Now you do not get not as much ATP as in an aerobic form, but you get enough to sustain yourself, and that is enough for them in that particular condition, okay, in the kind of lifestyle that they have. Okay. Okay, so this is the wine barrels and wine caskets that are kept. Okay, they are generally kept closed in an atmosphere of a lot more carbon dioxide instead of oxygen, because oxygen would spoil the taste of wine. You won't get alcohol. Okay, this is your large scale fermenter. Okay, this is just the payoff phase at each step. What is your net worth in the process at the end of glycolysis? Okay, this again tells you, this is one of the slides that you should know. It sort of tells you in a nutshell what happens to pyruvate, what is the intermediate step, what joins, what you get, kind of. It's like a summary. This is also a summary of your electron transport chain. Okay, so this is your overall summary that we walked through. Okay, now in this, mind you, sometimes in some of the pictures they would have written something like 32 ATP or 34 ATP, or total is 36 or 38 ATP, and this is a doubt which everyone has. Now you can get to be nitpicking for this whole thing. There is an actual calculation that has been done. Now this is for a biochem class which is studying deeper. They know that the calculation for 1 NADH is approximately 2.5 ATP. It is responsible for making 2.5, and 1 FADH is responsible for 1.5 ATP. And that is why you have a range of 36 to 38. Now, my class, I want you to remember 1 NADH is responsible for 3 ATP and 1 FADH2 is responsible for making of 2 ATP, which means our total ATP production out of every molecule of glucose broken down is 38. That's it. Okay. This is a highly theoretical situation, but let's go with that. Okay. This sums up the whole process of what is the electron transport chain and what is chemiosmosis. What I mean by that is your NADH starts on the inner mitochondrial membrane and starts to transport the electrons, throwing protons at every transfer, and there is a collection of protons outside because of that. At the end it stops, and oxygen is the terminal electron acceptor, and it is where the whole process stops, comes to a standstill, till there is a next one who will be transferring, and there is formation of water. This gradient of protons come rushing back in. So this particular process of electron transport chain and this process of chemiosmosis are two independent processes, but they are just aligned conveniently next to each other. Okay, and so together it's called oxidative phosphorylation. This is the account statement I already walked you through. You can even practice this looking at each step, and this is the answer key for it. And good luck. I hope this was helpful.